All right, good afternoon, everybody. Now I will be continuing with my the lesson that is antibodies, and in that I will be talking about the monoclonal antibodies. So let me just share my screen with you, and then we'll proceed with the class. So when it comes to monoclonal antibodies, it just means that so this is monoclonal antibodies and hybridoma technology. In this, you get to see that we have already seen the different kinds of immunoglobulins, and then we have seen the types of the functions of immunoglobulins in the last class. We have seen what T cells and B cells do and their activation, and then we have seen antigen antibody interactions, and then we have in this we have also seen the applications of antigen antibody interactions. Now we will be seeing the monoclonal antibodies and their application. So monoclonal antibodies are those antibodies which are identical in nature because they're only produced by one type of immune cell. So all the clones, that is all the cells that come from, they are similar in nature, identical you can say, from a single parent cell. Again, polyclonal antibodies are also present. They are derived from different cell lines, and they differ in their amino acid sequences. So again, you can see the differences between monoclonal and polyclonal antibodies. So many B cell clones produce polyclonal antibodies, whereas monoclonal antibodies are produced by one single B cell clone as it is. They bind to the different epitopes in the case of polyclonal antibodies, whereas in the case of monoclonal, they bind only to a single antigen. Then antibody classes they have a different type of antibody classes, and whereas monoclonal antibodies happen to be only from a single antibody class. They all bind to the same antigen binding site, in fact, and their cross reactivity is low. Whereas for antigen binding sites, in the case of polyclonal antibodies, they can bind with different kinds of antibodies with different antigen binding sites because of the presence of many epitopes that can be used, in fact, of the antigens. So the potential for cross reactivity is high. So if you say the basic definition is that it is a single type of antibody specifically used against a single antigen or a single epitope, a single kind of epitope. So it was first identified and produced by George Kohler and Susan Milston. They won the Nobel Prize in 1984 for the same, in fact. So antibody producing B lymphocytes with myeloma cells in vitro, that is, they created a hybridoma for that matter, and this was used against the antigens so as to recognize and work against that particular antigens. That is what single type of antibody, identical, directed against a specific epitope, that is antigen or antigenic determinant, produced by B cells of a single parent from a single hybridoma cell line, that is the myeloma cell line. So hybridoma cell means it's a fusion of a B cell lymphocyte with a myeloma cell, that is like a cancer cell, a myeloma cell. So some myeloma cells synthesize a single monoclonal antibodies naturally also. So production method is given over here. So when you get to see the establishment, this involves a few steps, that is immunization, cell fusion, selection of hybridomas, then culture of the same hybridoma cell, inoculation into a suitable host, and then that is followed by extraction, purification of the monoclonal antibodies. So this is a basic history that's been given about it. We have seen these. So production method, we will come to it. So what happens is, again, more or less, it's still largely dependent upon animal testing, requires immunization of the mouse. So we are talking about the first step, that is immunization to the animal, and usually a mouse with an appropriate antigen it is given. Then this antigen along with an adjuvant. Remember, we have studied about an adjuvant in the last class, which an adjuvant enhances the antibody production capacity. So adjuvants are non-specific potentiators of specific immune responses. Injections at multiple sites are given for the the mice. In fact, increased stimulation of B cell is seen, and in response to the antigen that is produced. This approaches of immune stimulated cells for synthesis of antibodies, so that maximum antibody production is seen against that particular antigen. Now, when the serum composition or the concentration of the antigen is optimal, that is maximum, animal is then sacrificed, spleen is removed aseptically, and then disrupted by mechanical or enzymatic methods, basically to release the cells. Then B cells of this mice are extracted from the spleen or spleen of the rat or rabbit, in fact. Then it is fused with tumor cells. Tissue culture cells produce the tumor cells or the myeloma cells. So there is a fusion of the antibody forming or the B cells with the myeloma cells. Then this extracted B cells added to a culture from the bone marrow for that matter. Then it's the fusion. This is used by a PEG medium, that is polyethylene glycol medium. And then once this is done, you have to select the hybridoma cells. So screening is done over here. So B lymphocytes having one particular gene, that is HRPRT1 gene, that is hypoxanthine guanine phosphoribosal transferase gene, codes for, this is the enzyme that is being coded for HPRT1 gene. In fact, this enzyme is involved in the synthesis of nucleotides from the hypoxanthine present in the culture medium. Therefore, B cells that grow in the medium having this hat, that is hypoxanthine aminopterin thymine media, but due to lack of this gene, myeloma cells do not produce this particular HGPTR gene. So it cannot grow in the hat medium. You've got a good picture of the same thing in your textbook for that matter. You can just have a look at it. So the myeloma cells fuse with another myeloma cells, they do not fuse, they all die in the hat medium as such. It is only the B cells that fuse with the myeloma cells, because B cells and B cells also do not fuse together. So they all die eventually. It is only the fusion of the B cells with the myeloma cells that is taking place. Then culture of the hybridoma cells take place in the suitable culture medium, or it is again supplemented with insulin, ethanol, different kinds of nutrients, amines, additional hormones for that matter. And then this is used in the production of the monoclonal antibodies. So you have different kinds of media such as DMEM, that is Dulbecose modified Eagle medium, and then IMDM, and you have the Roswell's Roswell Park 5, it is known as, so RPMI medium. Then you have Memorial Institute medium. All of these are the different kinds of media that produce the antibodies. Excuse me. Once this is done, antibody assay is done, and then you're going to have this inoculation of the hybridoma cell into the suitable host. So this is done when the hybridoma cells are taken, injected into the lab animals, again rat or rabbits for that matter. Then this is used for production of the monoclonal antibodies. These hybridoma cells, then they can be frozen and then they can be stored for future use. Again, extraction from the host animal, you can either sacrifice the animal or just and then you have to purify the monoclonal antibodies that are coming out from the animal. This is done by either chromatographic techniques such as ion exchange chromatographic techniques or antigen affinity chromatography, or you can do radioimmunoassay, or you can do immune precipitation. So these are the methods, or this is how you produce the monoclonal antibodies. The same thing has been explained over here. You can see immunized mouse splenocytes, myeloma cells, HGPRT without HGPRT and TK for that matter. So you have the minus sign over here, meaning that that gene is not present in the myeloma cells. Fusion with PEG, that is polyethylene glycol, microtiter plates with the HAT medium, that is hypoxanthine amino selection medium for that hypoxanthine aminopterin thymine HAT medium. Then after 1 to 2 days, you have to screen the culture with the supertatins. So you have to test the antigen specificity, antibody isotype, identify the positive wells, cloning of the hybridoma cells. Then you can freeze the cells, or again give it into the mice. And then from the mice sida, that is the SITs, or in the mice, you can remove this particular antibodies. So where are they applied? Where is the application of monoclonal antibodies? You can use monoclonal antibodies to identify tumors. So you can, by staining the particular tumor sections, diagnostic testing, that is, in the presence of that particular substance, you can either see for test for toxins, drugs, or hormones. Cancer therapy or immunotoxicology, that is again cancer cells having additional or new antigens on the surface, and normal cells can be compared together. So what happens is, if the monoclonal antibodies are given, they bind to the cancer cells, are isolated, so that it is further possible for us to identify the cancer specific monoclonal antibodies from them. Such antibodies from the can be used for identifying new drugs for cancer therapy. So this is known as immunotoxicology. And then you have autoimmune diseases, wherein you have in rheumatoid arthritis or ankylosing spondylitis, you have monoclonal antibodies having medicines appropriate for that. So infliximab, adalimumab, they are all effective in treating since they bind to the TNF alpha that is present in the the autoimmune diseases, and then they are they bind to the TNF alpha receptor, because of which this can be blocked in. So omalizumab is one particular medicine having monoclonal antibodies. It inhibits the human IgE, useful in treating moderate to severe allergic asthma. Then you have transplantation. So when there is an injection of bone marrow given, or for that matter any kind of transplantation, anti-T cell and monoclonal antibody is available. So what happens is, this removes the T lymphocytes that are present from the donor bone marrow, before the transplantation takes place. Then the anti-T cell monoclonal antibodies are injected in the bone marrow transplantation, so that this helps in preventing the rejection of the foreign organs, whatever that matter be, either the donor bone marrow, or the recipient when it accepts the foreign organs such as heart, kidneys for that matter, liver. So what happens is the body of the patient which has accepted these organs will not reject this particular donor organs. Monoclonal antibodies are also used for protein purification. So they are immobilized by coupling them with the cyanogen bromide activated on a chromatographic matrix, for example, such as saccharose. And then this is used for purification of the proteins. This protein mixture is sent through this chromatography column basically, so that you get purified proteins as such. This is because the monoclonal antibodies have this particular power for discrimination. So this is all about the monoclonal antibodies and their uses. As you can see, diagnostic purposes for during pregnancy, conception also, you can actually use them for highly specific diagnosis. Then they can detect the quality, quantity, presence, and they use for purification, like pure immunoprecipitation and affinity chromatography. Again, like I told you, graft rejection, self-tolerance, and alloreactivity. So then you have conjugated monoclonal antibody therapy. So this is all about monoclonal antibodies. So in the last lesson, in this lesson with the antibodies, we've actually seen about what exactly are antibodies. That is, they are immunoglobulins, which are soluble proteins made up of, produced by the B lymphocytes in the plasma cells in response to exposure to non-self antigens. Each antibody specifically is bound to a single antigen. They have a common structure, that is 4 peptide chains, 2 heavy chains, 2 identical light chains, and 2 identical heavy chains. This structure consists of 2 identical, and then they basically are glycoproteinaceous in nature. Several classes are there. Around 5 major classes of immunoglobulins are there: IgG, IgA, IgM, IgE, and IgD, differing in their size, their charges, the amino acid sequences that are present, and the carbohydrate content that is there. Interaction between antigens and antibodies specifically have the recognition of this antigenic epitope by the N-terminal variable region of the antibody molecule. Then, if at all there are any kind of antigen-antibody reactions that are taking place inside the cells, it is known as serological reactions. Then we have seen the monoclonal antibodies, their production, identified, and they are useful for identifying the specific antigenic determinant, that is the epitopes. So Kohler and Cesar Milstein had won this Nobel Prize in 1984 because they could help in the production of monoclonal antibodies in a larger scale for commercial, that is, using the hybridoma technology. So this term hybridoma is used to fuse the cells resulting due to the fusion of 2 types of cells, lymphocytes and tumor cells. Now, polyclonal antibodies are a mixture of heterogeneous antibodies and epitopes, basically used produced with different cells, that is the different B lymphocytes or B cell clones. Basically, they can recognize and bind to different epitopes in a single antigen. Please give me a moment. Right. Now, with this, we have finished our antibodies lesson. Now, we are going to have a look at the regulation of the immune functions. In this, we are going to learn about how the immune function is going to be regulated. And we are going to see basically major histocompatibility complexes, and what exactly is the complement system, what are cytokines, interferons, and what exactly is humoral and cell-mediated immunity, and summarize the whole lesson altogether. So when it comes to major histocompatibility complexes, we get to see that histocompatibility complexes are those which help in presenting the antigens to the cells. Just open. So they are surface proteins located on the cell membranes of the nucleated cells. Then they play an important work in identifying the antigen between the self and non-self, that is foreign. Then they are also useful for recognition in within the cell, that is intracellular recognition. And they majorly present the antigen to the the B lymphocytes and the T cell receptors. Then histo refers to the compatibility, refers to them living together in a harmonious manner. The MHC molecules recognize the T lymphocytes, and two types of MHC cells are there. They are the T helper cells are recognized by class 2 molecules, that is MHC, and T cells are recognized by class 1 MHC molecules. So it's a membrane attached protein that works on recognition of antigen between non-self and self body and the antigen presentations. So a brief history is also given basically. So my samples were used to identify the blood group antigen so as to designate it, and to the different kinds of groups wherein they are antigen presenting cells. So yes. So again, when it comes to this is one thing, the major histocompatibility complexes. Then they are again having extensive polymorphism, that is there of types, 4 types, 1, 2, 3, 4. We'll come back to that. And then we also have something known as the complement system in the body. So this is an effector system majorly seen in the humoral mediated immunity. So complement system has a lot of plasma proteins which are involved. In the absence of infection, what happens is these proteins are all over the body. They circulate in an inactive form. And if at all there is any kind of antigen present, or there is any kind of pathogen along with an antibody bound to it, then the complement system gets activated. Complement proteins interact with each other, forming several different pathways. There are 3 different complement pathways, basically 3 of so all of them will be activated, because of which finally the end result is that the pathogen is killed. The different byproducts, or the after the pathogen is killed, it is completely disintegrated, and then all the other parts are removed from the system as such. Then we have something known as the adaptive immunity. We have two types, that is cell mediated and the humoral immunity. So this generates antigen specific immune responses. So you have T cells, B cells that perform the humoral and the cell mediated immunity. So like I said, when it comes to the major histocompatibility complex, you have Peter Gorer, and he was one scientist who had seen that there are 4 groups of MHC molecules in the blood sample of the mice that identified the blood groups, the blood group antigens, which designated by class 1 to class 4 groups of the MHC. So George Snell, Jean Dosset, and Barry received this Nobel Prize in 1980 for their contribution for the discovery of the MHC molecules and understanding them in both humans and as well as in mice. So again, classes, like I told you, class 1 to 4 are there. Class 1 and 2 are worked in immunity. So class 1 MHC molecules express the glycoproteins, so nearly on all the nucleated cells, and each class 1 molecule encodes for a transmembrane glycoprotein of approximately around 43 kilodalton, referred to as the alpha or the heavy chain, comprises of 3 extracellular domains. I'll show you the picture if possible. The picture is going to come in a minute. Let us just finish this first. So, and then you have 3 extracellular domains, that is alpha 1, alpha 2, and alpha 3. Alpha is the heavy chain. Then alpha 3 is a highly conserved interacts with the glycoprotein CD8, which is present on the TC cells. So all the class 1 molecules expressed on the surface of a cell form non-covalent association with beta 2, that is a microglobulin, which is approximately around 12 kilodalton. This is encoded by a gene present on the other chromosomes. In humans, it is chromosome 15. And in the absence of beta 2 macroglobulin, this MHC class 1 molecule is not at all expressed on the cell membrane. So when it comes to class 2 molecules, you get to see that class 2 molecules encode for alpha and beta of approximately 35 kilodaltons and 28 kilodaltons. They, like class 1, are transmembrane glycoprotein molecules with cytoplasmic tails, extracellular immunoglobulin. You can see the picture over here, please. This is the class 1 MHC molecule. You have, This is the class 1 molecule. You will have alpha 1, alpha 2, and alpha 3. And this is the beta immunoglobulin chain. It is present in this form. And this is the disulfide bonds that are present over here. Class 2 is again, having alpha and beta chains basically. Alpha 1, alpha 2, beta 1, and beta 2. So 2 alpha chains and 2 beta chains that are present. And they are also members of this immunoglobulin superfamily. Glycoprotein CD4 is present on the cells, on the TH cells basically. This interacts with the variants of the class 2 molecules. They are present in all the conditions, so they are known as constitutive genes. And only on the specialized antigen presenting cells, that is. So APCs are classified as professional and non-professional antigen presenting cells also. These include the dendritic cells, macrophages, B cells, differ in one another in the mechanism of the antigen uptake. So dendritic cells are powerful, antigen presenting cells. They have high classes of class 2 MHC molecules. B cells have class 2 MHC molecules at low levels, possess the antigen specific surface receptors, and then they differ in the location, migration, behavior basically. So this is class 2 MHC molecules again that is present. These express the MHC genes constitutively only on the antigen presenting cells, like I told you, that is macrophages, dendritic cells, and B cells. MHC class 2 molecules also have two non-identical and non-covalently associated polypeptide genes. So you have alpha 1 and alpha 2, beta 1 and beta 2 chains. These two chains have amino acids ends on the surface, and a short transmembrane stretch is present over here, and an intracytoplasmic, that is within the cytoplasm you have the carboxy terminals over here, and both of them are MHC encoded and they are polymorphic in nature. Their alpha chain has alpha 1 and alpha 2 domains. Beta 1 and beta 2 domains are present on the beta chain. Then anti-allogenic sites also have that carry the determinants. They are formed between the alpha 1 and alpha beta 1 domains. So this is where the antigen binding cleft is present. In both MHC1 and MHC2, all the domains are stabilized by disulfide bridges except in the alpha 1 domain. You do not have any kind of disulfide bridge stabilizing this particular structure in the alpha 1 domain. Likewise, if you see over here, you have disulfide bridges all over the place, but you do not have it in the alpha 1 chain. So over here also you have they are expressed on all the nucleated cells, like I told you. And then the chain is encoded by the MHC genes. 3 globular domains, that is alpha 1, alpha 2, alpha 3 are present, and alpha 3 domain is non-covalently associated with the beta 2 microdomain or microglobulin. The beta 2 microglobulin is encoded by a gene which is present on another chromosome. So they are all again members of the Ig superfamily only, And beta 2 microglobin, like globin, the class 1 is not exactly expressed on the cell surface. If at all, there is no beta 2 microglobulin. So if at all there is a defective beta 2 microglobulin, then class 1 MHC is no antigen is not going to be expressed at all. So they have a deficiency of cytotoxic T cells. So again, greatest variable, this is the peptide binding cleft wherein, the antigen comes and binds over here. And greatest variability occurs in the alpha 1 and the alpha 2 sequences. They also bind to the T cell receptor of the CD8 T lymphocytes. So this is all about the MHC complexes. Now we will see about, I will send this particular PowerPoint with you, I will share it with you. So this is the, class 1 and class 2 molecules differences, where they are exactly found. And what they do basically, no invariant chains, has invariant chains, binding of CD8 cells in the case of MHC1, CD4 cells in the case of MHC2. So this is how they look. And then we have HLA antigen also. You can just have a, what do you call it? That HLA antigen is present in humans. So it's known as a human leukocytes antigen. And this is the major histocompatibility complex in the humans. So the differences in the humans and the mouse in the HLA antigen and the functions of it are given again. So I'm going to pause share right now, and I'm going to start with the classical pathway of the complement system basically. So when it comes to the complement system, please give me. Anyway, when it comes to the complement system, basically what happens is, it is a term that is used to describe the serum and cell surface proteins discovered by a scientist in the 1890s, that is, in late 1800s, in fact, by Jules Bordet. So they are glycoproteins in nature, synthesized by the liver, and that is the hepatocytes. And they're also produced by macrophages, monocytes, epithelial cells of the gastrointestinal and genitourinary tracts. They have around 5% of the serum globulins that are circulated in the serum, that is made up of complements. They actually are in the inactive form, so they, you can call it as proenzymes, and they are inactive until and unless they undergo proteolytic cleavage, after which they become the activated proteins. So because of proteolytic cleavage, what happens is, there's this inhibitory fragment that gets cleaved, and it gets to be removed, leading to the exposure of the active site. Now the complement reaction sequence starts with an enzyme cascade. In the presence of an antigen or an antibody antigen reaction, the complement system becomes activated. They interact with each other to form several different pathways of the complement activation. I'm just trying to see where this particular PowerPoint is. Just please give me a minute. So when it comes to the nomenclature of complements, you see that the complement components are designated by numerals C1 to C9, C1, C2, C3 up to C9, and they are also having letter symbols, that is uh, with the letters D, factor D, or trivial names such as homologous restriction factor. So the inactive proenzymes have a simple designation, that is either you can call it as C1 and C2. So they were named in the order of the discovery rather than the sequence of the reaction. So whatever was discovered first was named as C1, and what was discovered later was named as C3, like the later number C8, C9. So C9 was discovered the last, but that is not the order in which they start activating, getting they start working upon. So if you get to see, it is like the reaction sequence for the classical pathways C1, C2, C3 up to C9. Peptide fragments again formed by this activation of the components are designated by a small suffix, that is A, and the larger fragments as designated by the suffix B. Please give me a moment. I'm just like kind of seeing where this particular I had downloaded it, but I'm right now not able to find the same. So I'm just like taking from the net. Right. We'll just follow what is given in the text, in the net or in the textbook. Right. So when it comes to this complements, right, just follow this particular image. So this is the pathway that is there. B and what do you call it? A represent the complements, which are the fragments which are larger and smaller for that matter. For example, cleavage of C3 produces a small protein which is C3A, and the remaining larger fragment, that is C3B. So, and again by convention, the larger fraction is for the other factors is also designated as B. And for example, C2 is having a larger fragment, C2A, and the larger fragment binds to the target near the site of the activation, and the smaller fragments diffuse from the site, and they initiate the localized or the inflammatory responses. So there are complexes that are forming, that is C4B2A, C3B3B, etc. So the activated complement components are sometimes designated by a horizontal line, that is, for example, C2A. So when it comes to the classical pathway, this is activated by antigen-antibody complexes, either whenever there is an interaction with the immunoglobulin G or M. So they bind with the first component, that is C1. In addition, there is also an endotoxin which is present in the bacterial cell walls and certain viruses. This directly activates the classical pathway. In the classical pathway, it consists of the proteins recognition protein, that is C1q, and 6 subunits with globular heads associated with 2 molecules of C1s and C1r. So you have a C1r and a C1s along with the 6 subunits of C1q. Now, classical pathway is initiated when recognition of C1q is associated with proteases C1r and C1s. Either this recognizes a microbial surface or binds to directly the antibodies along with which are already bound to a pathogen. So this formation of the antigen-antibody complex, it is there in your textbook also. So this formation of this antigen antibody complex, what happens is it induces the change in the Fc portion of this immunoglobulin. So this causes the molecule to expose a binding site for the C1 component of the complement system. So again, serum which is present in the serum is a complex having C1q and two molecules each of C1r and C1s, which are together held in a complex which is known as C1qr2s2. And this all is stabilized by calcium ions. Please give me a moment. I'll just be back, please. Right. Now, so what happens is binding of the C1q to the Fc binding sites, this induces a conformational change in the C1r. This converts the C1r to an active site, that is C1r. So the star becomes C1r, because of which this has two substrates again. This cleaves to C1s to a similar C1s. So the star gets activated. So what happens is cleavage of the site leads to the active site which is being exposed. After this is done, C1, this cleaves the C1s to a similar active enzyme, that is C1s. This active enzyme C1s has two substrates, C4 and C2. So C4 is a glycoprotein. This has three polypeptide chains, which is alpha, beta, and gamma. And C4 is activated when this active C1s hydrolyzes a small fragment to C4A. And C4, from the amino terminus of the alpha chain. So this exposes a larger fragment, which is C4b. Now C4b attaches itself to the target surface in the presence of C1, and the C2 proenzymes then attaches to the exposed binding site on the C4b, wherein the C2 which is present, it is then cleaved by active C1s. Smaller fragment, that is C2B, just diffuses away. Now you have a complex that is formed, which is C4B2A, that is getting formed. And what happens is this formation of the C4B2A is called as the classical pathway that is being formed. Over here it is given as C4B2B basically, but it is actually C4B2A that is being formed. And this is done by C3 convertase enzyme. So what happens is this refers to the role in converting the C3 into an active form. So C3 convertases play an important role in this. You can see the same picture in your textbook also, in fact, for that matter. When it comes to an alternate pathway, what happens is spontaneous hydrolysis and activation of the complement C3 is, or component C3 is taking place. So, wherein, they bind directly to the microbial surface. So, this pathway involves 4 serum proteins, which is C3, factor B, factor D, and propyridine. 4 of them that are there. C3 is in 2 molecular forms. Native form, which is present in the serum, circulating it. And you also have an altered form, which is altered in the conformation, basically, which is actually a thiol-ester bond that can be hydrolyzed. This altered C3 will bind to another factor in the presence of magnesium ions. So, this factor B of the alternative pathway, as it is known as. Now, in the presence of factor D, which is a serine protease, factor B can be cleaved into BB, capital B, small b, and you have together, they form the C3B. That is, C3 is the component C3 along with the smaller fragment C3B to form the alternative pathway, that is C3B and capital B, small b. So, factor B, factor D is involved. You have then C3B, capital B, small b. This is the component which is getting formed. You also have this again goes into and works takes place in the inflammatory reactions, cell migration, activation of the same, etc. You also have a lectin pathway. In this, lectins are proteins which actually recognize and bind to carbohydrates, specific carbohydrate targets. This is initiated by soluble carbohydrate binding proteins such as the MBL, which is mannose binding lectin. You also have ficolins, which are also again carbohydrate binding proteins. So MBL is an acute phase protein which is actually produced during any kind of inflammatory reaction or response. So function is that it is acting in its function in the complementary pathway is similar to that of the C1q in the classical pathway. So structure also is similar in nature. Now this mannose binding lectin binds to particular carbohydrate structures which are present on the microbial surfaces. So microbial surfaces of what? Salmonella, Candida albicans, and you have Listeria, Cryptococcus neoformans, Neisseria strains. So bacterial strains, or for that matter yeast strains, all of them, any kind of the microbial strains having the carbohydrates, MBL goes and binds to it. So once MBL binds to the surface of the cell on the pathogen, then MBL associated serine proteases, that is MASPs, MASP1, MASP2 bind to the MBL now. MBL binds to the carbohydrates on the pathogens. MASP1, MASP2, they are the serine proteases, bind to the MBL, and then this MASP1 and 2 have structural similarity to that of the C1R and C1S proteins. So they mimic their activities. So what happens is this active complex is again cleaved, activation of C4 and C2 which is present inside the cells take place, and resulting you have a C4B2A complex called as a C3 convertase. So the C4B, that is the fragment B, and then 2A complex is actually called as a C3 convertase complex. Now these three complex pathways, that is either the classical or the lectin or the alternative pathway, converge. And at the central, you have the most important step wherein, the enzyme activity of the C3 convertases is generated. You have different kinds of C3 convertases present depending upon the kind of pathway, either the classical or the lectin or the alternative pathway that has been activated. But each is again, you have, it's a small subunit, multi-subunit protein having a specific protease activity. This cleaves the complements C3. So C3 convertase is bound covalently to the pathogen surface then, and then it gets to cleave the C3 to generate large amounts of C3b. C3 in the presence of convertase to give C3b. Now, C3b is the main effector molecule in the complement system. C3a, which is formed, is a small peptide that binds to the specific receptors. This leads to the inflammation or the inflammatory pathway to get activated. Now, cleavage of the C3 is the critical step in the complement activation. This leads to directly or indirectly all the effector activities or the subsequent activities of the complement system. Now, C3 can also, C3b, C3b can also bind to the C3 convertase, that is the enzyme produced by the classical and the lectin pathways. They again form another multi-subunit enzyme, which is C5 convertase. So, C3b, complement system can bind to C3 convertase to form C5 convertase, another multi-subunit enzyme. This cleaves the C5. This again releases highly inflammatory peptide, which is C5a. Then you have another C5b, which initiates the terminal pathway complement activation, wherein you have additional complement proteins interacting with the C5b that is being formed to form a membrane attack complex on the pathogen surface. So all of them form the membrane attack complex. This creates a pore in the cell membrane that leads to the cell lysis. So first step in the formation of the membrane attack complex is C6 binding to the C5b on the cell surface. Then C7, C8, C9 bind. They form a C5B678 complex, which is also known as the membrane attack complex. Now, when it comes to the functions of the complement system, they help in cell lysis. Whenever there's an immune response that is coming in, you have the lysis of cells, bacteria, viruses for that matter. Opsonization. This promotes phagocytosis of the particular antigens. Then binding to specific complement receptors on the cells of the immune system, triggering specific cell functions, inflammation, and secretion of immunoregulatory molecules. Immune clearance, which removes immune complexes from the circulation, deposit them in the spleen or the liver. So all this is taking place, these are the functions of the, complements. Now, let me start with cytokines. I'm stopping my share here. I'm going to again share my screen and go with cytokines. So in cytokines, basically they're small proteins in nature, less than 30 kilodaltons in fact, and they are released by various cells such as lymphocytes, monocytes, macrophages, etc. And they are usually produced in response to an active stimulus. So this induces response through the macrophages and binding to specific receptors, and cytokines usually act as at a short range, like endocrine or paracrine or autocrine, something similar to hormones. So they act like hormones basically in that is, they act on the cells in the vicinity, or they act on the same cells from which they are produced. So exceptions is that IL-1, that is interleukin-1, interleukin-6 can mediate release of the complement proteins from the liver. So this is one far-acting cytokines, that is interleukins 1 and 6. Term cytokine is actually meaning that it encompasses those cytokines secreted by lymphocytes, substances known as lymphokines. These are secreted by monocytes and macrophages. So earlier it was known as monokines, and many cytokines are actually referred to as interleukins. That is, they are secreted by some leukocytes and they act upon other leukocytes. So potential, like I told you, their function is they, they are like, either local, that is paracrine, autocrine or endocrine, highly interactive, that is they're pleiotropic or redundant or synergistic. We'll come to that later. And this is how the cytokines are produced. So again, they have a high affinity. Please give me a moment. So they have a high affinity towards, just a second, I'm trying to get in that particular, right. They have high affinity with the receptors, and they induce the, they exhibit induction of some of the following functions for that matter: cascade pathways and redundancy, pleiotropy. That is, when it is in a cascade pathway, what it means is that one cytokine, if it gets activated on a target cell, it induces the cell to produce more than one other cytokines. This in turn produces other cytokines. So it is a cascade pathway. Redundancy means that it is two or more cytokines work in the same manner and mediate a similar function. Pleiotropic means that a given cytokine has different biological effects on different target cells. If you see activated T cells, target cell over here is target activation, proliferation, differentiation of B cell. But interleukin 4, which is the cytokine on a thymocyte, helps in proliferation, and in a mast cell it also helps in proliferation. Whereas if interleukin 4 acts on a B cell, it helps in proliferation, differentiation, and activation as such. And synergistic in nature, that is, for example, two cytokines together form or act on one cell. You can see redundancy example also here. Interleukin 2, 4, 5 all act on the same cell, that is, and induce the same function, that is proliferation of the B cells. And they can also be antagonistic in nature, that is, if at all there are two cytokines, they can go and they inhibit the effect of another cytokine. So whenever interleukin 4 is acting on the B cell, then this, what happens is, it causes activation of proliferation, differentiation. But when will that proliferation stop? That is when INF gamma, another cytokine, goes, acts on interleukin 4, and this stops the production of activation of the IL-4, so that proliferation of B lymphocytes or B cells stop. So this is all about the functional features of the cytokine families. And again, they are target specific, that is, they bind to only a specific receptor or a specific cell membrane of the target cell. This triggers signal transduction pathway. Do I have a picture of it? Let me see. So this is how it is. This is how it is again. You can see again, they are helpful in immunoglobulin and humoral as well as cell-mediated response. We'll come back to it in the next class. Let me finish cytokines and then you can leave. So when it comes to the target specific, it binds to specific receptors on the cell membrane of the target cell, triggers the signal transduction pathway, so that ultimately in a signal transduction pathway, what happens is gene expression is altered. When it comes to the action of the cytokines, it can be autocrine, or it can be merocrine or endocrine also. That is, in an autocrine manner, they act on the same cell. In a paracrine manner, in the nearby cells. Endocrine manner, in the distant cells. Interleukin 4, interleukin 6, for that matter, is the example. Then chemotaxis. So cytokines released by the tissues, they are membranes. They attract other they belong to this family called as the chemoattractant cytokines or the chemokines. These small proteins induce the directed chemotaxis in nearby cells, making them more responsible, resulting in the movement of the cells towards the sources of the chemokines. So when it comes to the biological functions of the cytokines, you can see that they are helping in the regulation of hematopoiesis, healing of the wounds, induction of inflammatory responses, stimulate development of cell and humoral responses, and control of cellular proliferation and differentiation. And there are different kinds of cytokine families that are present basically. So 5 families. And if you get to see, the 5 families include the hematopoietic family, interferon family, chemokine family, you have the tumor and necrosis family, and you also have the hematopoietin family. Not exactly given in this, but you can actually see it in your textbook. Immunoglobulin superfamily plays a role in inflammation, and it induces receptors for interleukins 1 m, that is macrophages, colony stimulating factors, c-kit, and interleukins 18. Interferon family, for that matter, is multimelic receptors. They are composed of heterologous subunits. Basically, these are the receptor families. You can just have a look at it. Interferon family is this. This includes the INF alpha, beta, gamma, and tissue factors, including then IL-10, IL-22, and tissue factors. So when it comes to the chemokine receptors, they are G protein coupled receptors with 7 transmembrane structures, and they couple the G protein, and this triggers the signal transduction pathway. Then you have the tumor necrosis factors, that is the TNF family over here. In this TNF family, they have a cysteine-rich domain. All of these are cysteine proteins, and they are formed of 3 disulfide bonds surrounding a core motif, and this is associated with procaspases, and through that is through adapter proteins, FADD, triad, etc. This helps in the caspase pathway that is being triggered, so that this plays an important role in the apoptosis, that is programmed cell death. Then you have the hematopoietin family over here. Let me just see. I think I have a picture of it. This is the hematopoietic family. Again, a majority of this is the cytokine receptors. They belong to this class. These receptors have certain motifs in the extracellular domain, basically, that is the amino acid domain. They lack an intrinsic protein, that is tyrosine kinase activity is not present in this particular proteins, basically. This family includes IL-2 receptors, that is the beta subunit, IL-3, IL-4, 5, 6, 7, 9, interleukins 11, 12, then GM-CSF, then granulocyte, colony stimulating factors, then LIF, CNTF, all of these are receptors that are present in the hematopoietin family. They are also receptors for thrombopoietin, prolactin, and growth hormones. So this is there. Then you have interferons in the humoral and the cell-mediated activity. Please give me one moment. All right, then what I will do is I will stop this particular topic for today. Tomorrow, I will continue with the interferons, the humoral and the cell-mediated immunities, summarize this particular lesson, and then go for immune system in health and disease. That is your hypersensitivities, autoimmunities, the immunodeficiencies that are present. All right, so thank you so much.